Hi everyone. My name is Bill. I'm from the University of Chicago, and I'm here with my colleague Kang Yu, who is from St. Jude. And today we're going to talk to you about sequence reads analysis with GDC 2.0. And I'm going to give you a short introduction and just give you an agenda for what we're going to be talking about today. So first is going to be a short introduction about the two tools that are going to be demonstrated. These are the band slicing tool and the sequence reads tool. That'll be followed by two demos by Kong Yu. He will show us the band slicing tool and the sequence reads tool. And at the very end, we will talk about some resources for support for these tools and have some Q&A. So if you have any questions during the webinar, feel free to type your questions into the Q&A panel. If you don't see that right away, I would recommend clicking on the three dot button and you'll see a Q&A button there. We'll definitely see the Q&A in there. If you type it in the chat, we might not see it, but sometimes we'll check that just to make sure we're all covered. So Q&A panel, we'll answer those at the end. So just to start out with a reintroduction to the GDC 2.0 workflow, here is a flow chart showing how it works. And it starts with building a cohort, which is a group of cases based on whatever cancer you're interested in or whatever attributes you would like those cases to have. And then you have the opportunity to download data. But today we're going to talk about analyzing the cohort with two specific tools from the Analysis Center. One is the BAM slicing download, which is the top left here. And the other one is the sequence reads, which is more bottom to the right here. These may not be exactly where they're placed on your analysis tool cards, but you will see the cards there. So to first give an introduction to the GDC BAM slicing tool, this tool allows users to save space by downloading a specific region of any GDC aligned BAM file. That is one of the main functions here. You won't have to download the entirety of a BAM file. Only a specific region can be downloaded, and those regions can be custom genomic positions, so coordinates for a particular chromosome can be used. Genes, just specific genes can be used, or entire chromosomes. So all of those are options to download with the BAM slicing tool. The GDC BAM slicing function is also available through the GDC API. If you're interested, we're not going to talk about that today, but if you're interested in that, see the June 2023 webinar in which I talked about GDC BAM slicing and how that can be done. So you may be asking, what do I need to use the BAM slicing tool? The first thing you'll need, and that actually applies to everything in this webinar, is dbGaP access to the project that has the BAM. So if you're looking to download from TCGA, you need access to TCGA, and that applies to any project. You'll also want to have a specific position in the genome to download, so that can be coordinates, a specific mutation, a specific gene, or an entire chromosome or several entire chromosomes. Those are all options for BAM slicing. Because the GDC 2.0 is cohort-based, you will need an active cohort that includes the cases associated with the BAM. If you just have a list of BAM IDs, that's fine too. You can use all of the cases in the GDC as your active cohort. And then the last thing I want to mention is in the GDC RNA-seq pipeline, there are three BAMs that are created. There's the genomic, the chimeric, and the transcriptome BAM. So the transcriptome BAM is sorted in a different way, so it can't be used with GDC BAM slicing. But the genomic and the chimeric one both can be used. So because those are sorted in a standard way, you can. The transcriptome BAM, the coordinates are weird, so you can't download exactly from there. So now I'm going to talk about the GDC sequence reads tool and give a short introduction to that. This tool allows users to visualize reads as they are aligned to the reference genome. Think of it like the BAM slicing tool, but you are looking at the slice rather than downloading it to your computer. This can be used to investigate, any specific reads you're interested in, or genomic positions. Maybe you found something interesting and you want to see why a variant caller made that specific call. This can all be visually investigated using this tool. Also, annotations, visualizations, and zoom levels are all customizable in using the tool. So those features will be demonstrated by Kangyu in a bit. And then whatever I said, this is kind of like visualizing a BAM slice, and whatever you are looking at in the tool can be actually downloaded um, using the tool. So that is an option there. To use a sequence tool, like the other one, you will need dbGaP access to the project that has the BAM. And something I want to mention that Kongyu will likely give more details about is that everything you're going to see in this presentation is completely open access. All of the BAMs that are currently in the GDC are controlled access. So we can't show you what's on production, but St. Jude has some test data that they can show us that is open access. Um, like the um, like the BAM slicing tool, you will need a specific position in the genome to look at, or a specific region. This can be coordinates, mutations, genes. Entire chromosomes might be a bit too zoomed out to do anything with, but I know that if there are few enough reads, you can look at that. And then, like the other one, you'll need an active cohort that overlaps your BAM. And this has the same limitations with the transcriptome RNA-seq BAM. So you can't use that, but you will always have the option to visualize the RNA-seq reads that are aligned to the genome. So all that being said, it's time for the demo. So I will stop sharing my screen and pass it to Kongyu.
Thank you, Bill. Share my screen. Yeah, so I will demonstrate the basic use of the BAM slicing download tool and the sequence read visualization tool. And as Bill just said, because all GDC BAM files are controlled access, in this demonstration, we'll be only using non-GDC open access BAM files. So to access the BAM tools from the GDC data portal homepage, we click this analysis center. And the BAM slicing download tool and the sequence read visualization tool will be shown in this analysis tools section. And to open any tool, we just need to click this play button in the up right corner of the card. And I will start with this BAM slicing download tool. And the GDC BAM slicing download tool allows us to download a specific region of a BAM file instead of the entire file. After the tool is launched, first we need to select a BAM file. We could do this by searching for the file name, file UUID, case ID, or case UUID, or we could just browse the first 1000 available BAM files and select a BAM file from here. And I will just select a BAM file from here. And now the BAM file is loading. It might take a while. And yeah, now the BAM file is downloading. We can see this green check mark, which indicates that the BAM file has been loaded. And we can also see this two-column table, which displays some brief information about the BAM file, including the entity ID, experimental strategy, tissue type, tumor descriptor, and the size of this BAM file. And below the small table, we have this bigger table that allows us to choose where to slice the BAM file. And this table has 3 tabs. The variant tabs displays all the variants associated with the case. And this gene or position tab allows us to enter a gene, a position, a SNP, or variant that we want to use to slice the BAM. And lastly, this unmapped arrays tab allows us to download only the unmapped arrays from the BAM file. And here I'll just use TP53. And after we select where we want to slice the BAM, we click this submit button here, and then the sliced BAM file will be downloaded. Okay, so next I will move on to the sequence trees visualization tool. And similar to this BAM slicing tool, after the tool is launched, we first need to select a BAM file by searching with the file name, file UUID, case ID, or case UUID. Or we could just browse the first 1000 available BAM files and select one from here. And again, I will just use one BAM file from here. And after the BAM file is loaded, again, we see this small table displaying some brief information about the BAM file. And this bigger table here allows us to select which region of the BAM file we want to visualize. And this table has two tabs. This variant tab displays all the variants associated with this case. And the gene or position tab again allows us to enter gene, position, SNP, or variant. And I will again use TP53 here. And then we click this submit button. And now the protein paint BAM track is displayed in the genome browser. And here I want to mention again that we are using non-GDC open access BAM file in this demonstration. And our BAM track is designed with features that specifically useful for cancer research. A paper describing this BAM track has been published in the Journal of Bioinformatics. The paper has a tutorial and it covers more features in more details than what we are going to show in this demo today. So this in this main visualization screen, we can see it consists of a toolbar at the top and a reference genome sequence and a gene model track and a BAM track, which is composed of a pile-up plot and a read alignment plot. So in the toolbar section, this coordinates field shows the coordinates of the region that is currently displayed. And we can actually change the coordinates here to change the region we want to visualize. And this in and out 2, out 10, out 50 buttons here, they allow us to zoom in or zoom out of the current region. And another way to zoom in is by dragging on the genomic ruler to select a region to zoom into. And we could also click and drag the graph left and right while maintaining the same level of zoom. And below this toolbar is the reference genome. So if we were zoomed out, we probably won't be able to see the sequence in this reference genome. So now we are zoomed in, so we'll be able to see the sequence. Let me zoom out. And below this reference genome sequence is the gene model track from gene code, and below which we have a pile-up plot, which displays the total read depths and each nucleotide position. And the green color here represents reference oligonucleotides. And the red, orange, light blue, and dark blue each represents mismatched A, T, C, or G nucleotide. And below this pile-up plot, we have the read alignment plot. And here we have about 1,000 reads. In order to show all these reads in one screen, these reads are rendered in global view, with each read represented as a very thin line. In order to see the actual read sequence for the part of the reads you want to look at, we just click on the part of the reads, and then we will be switching from the visualization of the full set of the reads into visualization of only a subset of the reads. And this green scroll bar here will only show up after we switch to this partial view. And it gives us a general idea which subset of all reads we are looking at. And in this partial view, each read is represented as one row. And if we zoom in, we'll be able to see the actual read sequence. And to go back to the global view from this partial view, we could just click on the gray area of the scroll bar region. And then we'll be switching from the partial view to the global view, which has all the reads shown in one screen. And if we want to look at another subset of the reads, like this area, we can just click here. I'll be switching, and then we'll be switching from the global view to the partial view again. And to view the read above or below this area, we could scroll up or scroll down with this green scroll bar. And the top and bottom of this green scroll bar can also be adjusted to display more or less reads on the screen. And here we can see that these nucleotides are rendered in different backgrounds, colors, and lightness. And we can find a detailed legend. So we have a color scale legend here. And the lightness of these colors represents the Fred base pair quality score. So the dark color will represent like this dark gray color will represent higher quality score, and this lighter color represents a lower quality score. And the gray color here represents that the read is properly aligned, and the red here represents mismatch, and the blue here represents a soft click. And if we scroll up, we can see that the single nucleotide insertion is rendered as a sign color letter inserted between the two reference nucleotides. And if we scroll down, we can see that the deletion is rendered as a black line covering the span of the deleted bases. And next, let me go back to the global view, and I want to next demonstrate how this read sequence visualization tool supports improved on-the-fly variant genotyping. So for any given variants like this deletion mutation shown here, the tool will classify reads into different groups. Like for this 18BP deletion mutation in TP53, the reads are classified into a group of reads that supports the alternative allele. and a group of reads that supports the reference allele, and a group of reads that do not completely match with either the reference or the alternative allele. And this group only exists if we choose to use strict mode to classify the reads. And we can see that this is mostly caused by the low sequencing quality. So we have a lot of nucleotides with these lighter shades, which represent a lower Fred base pair quality score. And lastly, we have this ambiguous group, which contains the reads that have equal allele similarity to both the reference and the alternative allele. And if we go back to the reads that support the alternative allele, move it to the left a little bit, we can see that these black lines that representing deletion, they directly match with the given variant. But for these reads that carry a long soft clip at the right end or at the left end, or for these reads that carries these mismatches and insertions, it is now obvious that they are supporting the deletion variant. So to further investigate whether these reads actually support the deletion variant, we could switch to the partial view to see only the portion of the reads we want to look at. And then if we click on a specific read, like this one, a read information panel will be shown to display the details about this specific read. And from this panel, we can click this button here to align this read to the alternative allele. So here, this second alignment is aligning the read against the mutant allele. And we can see that it's a perfect match, showing that this read is indeed supporting the deletion variant. And next, I will switch to the left. I will switch to some other BAM track examples to showcase additional features. So here in this first example, this example is a focal genomic deletion targeting one exon of the gene. And I will show a few features that support the deletion. So first of all, from this pile-up plot, we can see a dip of read depth. And from both ends of this deletion, we see the soft clips. If we zoom in, we can see clean-cut soft clips, which suggesting a DNA breakpoint. And let me zoom out. But again. And in this config button, we have a detailed legend showing the discordant reads. So from here, we can see that from here, we can see these legends. And also from here, we can switch to paired mode instead of a single mode we are using. So if we select here, then this read will be shown in paired mode. And we can see that in the paired mode, when the coordinates of the read pair do not overlap, the read pair will be plotted in one single row and will be joined by a gray dotted line. And when the coordinates of the read pair overlap, the read pair will be plotted in separate rows with a blue line indicating the overlap. Yeah, zoom out again. And for this green reads, and from the configure panel, we can see that it's read pairs that have the wrong insert size. And we can see a lot of these green read pairs spanning this region, suggesting a deletion from this region. And now I move on to the second example. And this example, we have a 8BP insertion in the exon. So if more than one nucleotide is inserted, a number will be shown between the two reference nucleotides, indicating the number of inserted nucleotides. And if we add in the variant, this 8BP deletion, we can see that all these reads with the 8BP insertion actually support this 8BP variant. Let me go back to the example. And this example is from RNA-seq data. So we could see these spliced reads and the splicing over the intron. And the last example I want to show is this genomic. This example shows a genomic translocation. And here we can see that the soft clip reads are aligned. And if we zoom in.
No, Tony, you are showing the wrong one. You should switch to the.
Oh yeah, sorry. This is the 8BP deletion. Yeah, refresh. So this example shows genomic translocation. Here we can see that the soft clip reads are aligned. And if we zoom in, we could see that the sequence of these soft clip reads are identical, suggesting the presence of a breakpoint. Let me zoom out. Zoom out again. And the reads in orange are discordant reads with a mate mapped to a different chromosome. If we click on one of these orange reads, we can see that its mate is mapped to chromosome 21, where the gene RUX1 is. And if we zoom out enough, we could see that the chromosome where the mate of the read mapped to is printed on top of the read. And this brings me to the end of my demonstration. I'll pass it over to Bill to continue. Thank you.
Thanks, Kangyu. Another good demo, another opportunity to show off some of GDC's capabilities. Let me go ahead and paste some of these. I'm about to show you all some information, and apologies for the preliminary message before. I was trying to insert a return into the chat, but that didn't quite work in WebEx. But let me go ahead and share my screen. And I don't think I'm going to return, or I'm not going to need to return to PowerPoint just quite yet, because most of the things I'm going to show you are going to be on just their browser. So the first thing I want to bring to your attention, which is the first link that I sent in the chat, is the bioinformatics publication that was generated by St. Jude. This is regarding the these two tools. So if you're interested in how that was developed, feel free to give this a read. I see a couple of the authors in the attendees. So great job, everyone. The next thing I want to point you to is the GDC documentation website. Now, if you're interested in those two tools, those we have users guides to show all of the different functionalities that we're talked about here. So if you go to docs.gdc.cancer.gov and you click on Data Portal, all of the different apps each have their own um, page here. So if you're interested in BAM slicing, you click on BAM slicing, and we have a whole user's guide on that. And same with sequence reads. So yeah, visit our documentation site if you're interested in that. The last thing I want to show is our GDC website, gdc.cancer.gov. And what you'd be probably interested in here is going to the support menu here and first clicking on GDC webinars. This is likely where you found this webinar, and the recording of this webinar along with the presentation will be available here along with all the past webinars. So when I talked about the June 2023 webinar, you should be able to see it right about here. So if you click here, you could watch that webinar, the YouTube video. There we go. YouTube might be a little slow today. Anyways, the other things I want to show you are the tutorial videos, which you can get to here. And this is just an introduction to all the different analysis tools. So these aren't webinars. These are videos that were produced to show off GDC 2.0. So these two analysis tools do have their own your BAM slicing and sequence reads. So you can watch videos on those that aren't this webinar, but when this webinar comes out, you'll be able to watch it. And then lastly, I'll go back. Oh, if you go to support, I'd recommend joining the GDC user listserv. There is any announcements that come from the GDC about data releases or software releases will come from here. And if you have any questions about anything that happened today, or anything GDC related, contact the GDC help desk. And that email address is let me go. So we have it big here. Can be found at support@nci-gdc.datacommons.io. So that is the end of the presentation, and we will take any questions. Thank you, everyone. I don't see any questions yet. Oh, here's one.
Yeah.
All right, I'll go ahead and read it. Thank you for the introduction of these tools. I have two questions. One, if I input a single position into the BAM slicing tool, will it download all the overlapping reads? Two, is it possible to use the BAM slicing tool in a programmatic way through an API? And first, I will say that the answer for two is yes. I'd recommend watching our webinar from June 2023, and that has instructions on how to download things via the API. Also, we have that in our documentation site under the API section. So if you were to go to API, BAM slicing, all the instructions are here. But I'll leave the first question to Kang Yu or Shen.
Yeah, it will allow you to download all the reads that overlap with your input region. It is correct.
Awesome, thanks, Shen. And yeah, we will. I guess we'll wait for any other questions that come in for a couple of minutes.
So Bill, looks like there are no other questions, and thanks to you and Kongyu for. I'm sorry, there is a question. What is the website to watch previous webinar videos? And so, Bill, do you want to go to the GDC website
Yeah,
webinar
sure.
page?
So if you just go to gdc.cancer.gov, and you go up to, sorry, some of those WebEx windows are in the way here. Go to support, and then click on GDC webinars. You'll see all of them here. And you'll see a preview show up here, so you can click that and watch it on YouTube, or you can click on read more. And you have a bigger screen, or you could always just click the YouTube link and watch it there. So all of the webinars are located at this site here. And then I saw another question. I wanted to download data for CLL. However, I did not see any. What does CLL stand for? Apologies, I'm not familiar with CLL. So while you type that in, can any of you be contacted for further discussion about GDC? So to answer that question first, if you go to Contact GDC Help Desk, and let me click on support here. I think it brings it up. Right here, support at nci-gdc.datacommons.io. This is a team that I manage, and sometimes I will be answering these questions, so you will hear directly from me if you email this, or one of my colleagues. So we will be, you can contact us through this help desk. But in terms of chronic lymphocytic leukemia, I am not sure. I mean, the possibility is that simply GDC doesn't have any projects with that yet, but I don't know all of the cancers we have off the top of my head. So if you search around primary diagnosis, I think that's the field that would have it, but I am not sure at the moment. But if you contact the help desk, we'll definitely look more in depth for you. Well, yeah, thank you all so much for joining us. If there aren't any more questions, I guess we'll end the webinar there. Yeah, Sharon, do you have anything else for the group?
No, I was just looking at our cohort builder by primary site and looking at there's like we have chronic myelomonocyte leukemia and chronic neutrophilic leukemia.
Okay, gotcha. All right. Yeah, thanks everyone so much for joining. And I guess I will stop sharing my screen. And yeah, if you have any questions, this is your last chance. Type it in. Awesome. Then yeah, hope you all have a good rest of your day.
Thanks, everybody.